Well, good morning, everybody. This is Giuseppe Planas from the International Pacific Halibut Commission. You're listening to the seminar series, and today we have the pleasure of having Dr. Wes Larson speaking for us after a brief interruption in our seminar series, and we're going to restart that, and hopefully we'll be active in 2021. So Wes is currently he's a program manager at the genetics program at the Auke lab that's part of the Alaska Fisheries Science Center in Juneau in Alaska. And Wes got his PhD from the University of Washington at the School of Aquatic and Fishery Science. During his PhD, he did extensive work on Pacific salmon population genomics. Then he moved as assistant unit leader of the USGS Wisconsin Co-op Fishery Research Unit. It's part of the University of Wisconsin. And from there, he's now in Juneau, as I said. So it's really exciting to have Wes. He's going to speak on a topic that is of tremendous interest for us. So thanks a lot, Wes, and go ahead.
Cool. Well, thanks for inviting me. Joseph, can you guys all hear me?
Yeah.
All right. And I'm going to do the screen share here. Look good. Slide view and all that? All right. Perfect. Well, thanks so much for attending, everyone. Today I'm going to kind
of... if anybody has any questions, they can type it in the questions tab, and I will read them at the end of the presentation. Sorry about that.
Oh, no worries. Perfect. All right, I'm going to shut my blinds because it's actually sunny in Juneau today. No way. Can't believe I'm inside. All right. So I'm going to start out today with a little bit of kind of some background on conservation genetics and the transition from genetics to genomics, and then talk about a couple examples of using genetics and kind of adaptive markers to understand Pacific salmon. And then I'm just going to finish up with a little kind of quick overview of some of our recent eDNA work. I know IPHC is interested in eDNA, and we sure are as well. So I figured just kind of a quick update on what we can do and where we've come so far would be nice to show. So all right, let's get into it. And I promise there'll be more fish pictures later, but the first few slides are some kind of some important background knowledge. So just to kind of keep things in context here, my view, one of the primary goals of conservation genetics is to conserve diversity. And, one of the seminal papers kind of on this topic came out in the early 90s by Robin Waples, and it was kind of defining what an ESU is, or an evolutionary significant unit. There was a lot of work at this time defining different units of salmon in the Columbia. Kind of a huge boom in this sort of research area, and Robin put out some of the some of the really seminal papers on this. And so as far as defining an ESU, there's kind of two major components of it here. There's trying to identify substantially reproductively isolated populations and populations that represent important components of the evolutionary legacy of species. And kind of just one thing that's really important to keep in mind here is that plenty of studies have shown that losing diversity jeopardizes species viability. So this within species diversity, this diversity of different life histories and ecotypes and populations really contributes to kind of the long-term sustainability and resilience of species. Okay, so how do we actually define these units? And one of the major ways, not the only way, definitely things like phenotypic evidence and ecological evidence are very important, but one of the major ways we define ESUs is using genetic markers, also called loci, to estimate differentiation and diversity. And this is from another really good paper by Robin called Separating the Wheat from the Chaff, which focuses on trying to identify stock structure in marine populations with low genetic differentiation, which is still a really good read. So we use genetic markers to estimate differentiation. We determine if the populations or stocks are significantly reproductively isolated based on this genetic data, and then we design management strategies to protect these genetically differentiated stocks. And so the next thing I kind of want to get into here is neutral versus adaptive loci. So neutral loci, which has been kind of the basis of these kind of ESU population designation stock structure analyses for the last 40 years plus. And neutral loci reflect neutral processes such as migration and genetic drift. And they can help us understand reproductive isolation, which is kind of the first tenet of that ESU definition. On the other hand, adaptive loci reflect selection. They diverge more quickly generally than neutral loci, and they represent or at least link to functional diversity. And so adaptive loci kind of work more in the second part of the ESU definition, in this evolutionary legacy part. So it's actually identifying kind of the functional differences between different populations. And so identification of ESUs has essentially used neutral loci for decades, because adaptive loci are very difficult to discover. So there's only a very small portion of the genome that actually codes, for different genes. And so actually trying to, survey these adaptive loci has been very difficult because we've only been able to genotype a few loci. And this is just an example of a really common adaptive locus in salmon, which is the major histopathology you can see. That most loci in this system show very low differentiation, but the MHC is extremely highly differentiated. So it's a really nice, and one of our first examples of adaptive loci in salmon. So like I said, for the past 40 years or so, we've been working in the kind of the realm of genetics, which is using 10 to 100 markers to look at neutral population structure, genetic diversity, and parentage. But now we've kind of transitioned to genomics, which is facilitated by these advances in sequencing technology. And just to put it in context, with genetics, with like Sanger sequencing, which is a kind of traditional genetic technique, you could maybe generate, hundreds of sequences a day with a single machine. And with sequencing technology now, we're up into the billions of sequences per day. So it's, it's orders of magnitude more data. So instead of 10 to 100 markers, you're talking about thousands to millions of markers. And this is extremely increased resolution for, this provides extremely increased resolution for kind of understanding neutral population structure and genetic diversity. But it also allows us to answer new questions related to selection, adaptation, trait mapping, and fitness-related genes. So in other words, it allows us to actually start looking at those adaptive loci rather than just focusing on the neutral loci. And so here's just a quick example of the importance of adaptive loci. And this one's from Atlantic cod, which is a really nice example. So Atlantic cod, there's kind of these two major ecotypes, migratory ecotypes and stationary ecotypes. They're also called fjord versus open ocean ecotypes. Essentially, the migratory ecotype kind of moves around deeper water, and the stationary ecotype, stays more near shore in these fjords. And if you actually look across the genome, there's very little differentiation at most chromosomes, but there's actually one major chromosome that shows extremely high differentiation. It's actually because a huge piece of the chromosome is an inversion. We'll get into that type of thing later. But the main point here is that most of the genome shows these patterns of neutral structure and no real differentiation among these ecotypes. But there's kind of one chromosome that's showing extremely large adaptive differences among the ecotypes. And it turns out that these ecotypes actually respond very differently to fishing pressure. So this is from a recent paper, a really cool one. And essentially what it shows, we don't need to get into the details of the graph, but basically the different ecotypes responded differently to fishing. So the LG1 linkage group 1 homozygous inverted ecotype was more affected by fishing than the other ecotype. And this is pretty, there's been some other studies that don't point it out quite as cleanly, but essentially it's pretty easy to think about in concept where, if something's near shore, it's in a fjord environment where it's concentrated, you might be able to, hammer on it pretty easily compared to something that moves around, has more of kind of a, more connectivity among small kind of subpopulations and things like that. So this is just a really nice example of showing that these different ecotypes with adaptive differences, but not really neutral differences, can be affected very differently by fishing pressure. So again, the population dynamics of these ecotypes differ substantially, and this ecotype specific management is really necessary. So just to kind of go back, if we looked at this based on a neutral structure standpoint, we wouldn't have defined these as different. But based on these adaptive markers, we can see that they really are different. And it turns out that there's a functional differences in these ecotypes that manifest as different responses to the fishery. Okay, and I wanted to revisit this wheat from the chat paper again because this is really important stuff. So adaptive diversity is really important in marine species, and this is a really nice kind of summary of marine species here from Robin. So in many marine species, high levels of gene flow ensure that the genetic signal from population differentiation is weak. As a consequence, various errors associated with estimating population genetic parameters might be normally safely ignored. Kind of assume greater importance. So what this is saying is, like, in salmon, because of natal homing, you know, it's often pretty easy to define kind of at least broad scale population structure. But that's definitely not the case in a lot of marine organisms. And so adaptive loci can be really, really useful for disentangling this population structure in marine organisms. And marine organisms, there's obviously few barriers to gene flow, which actually leads to this clustered genomic architectures of adaptive divergence, kind of like we see in the cod. And so essentially what that means is that because there's no like actual physical barriers to gene flow, these kind of really conserved architectures of genomic of adaptive divergence seem to evolve in marine species. So essentially it's really easy, or it's really important to look for them. And then also in marine species, low neutral structure actually makes adaptation easier to detect. So kind of just the main take home from this slide are, neutral loci have limited utility in many marine species, and adaptive loci can substantially improve resolution and provide information on evolutionary significance. So that ESU part 2. In other words, marine species are, I think, a really important place to look for adaptive loci. They have very low neutral structure, so your kind of normal genetic analyses may not be very useful, and they kind of have really good opportunities to evolve these adaptive genetic architectures. And so I guess it's kind of my advocation for making sure that we look at adaptive loci in marine species. Okay, so now because I just started working on marine species again, most of the rest of the talk is going to be salmon. But hopefully I've convinced you guys that adaptive diversity is important. And so where I'm going to go here is kind of trying to figure out how we can identify it and use it to improve management. Okay, so that was a lot of marine species, what you guys work on, what I largely work on now. But my background is in salmon, so I'm going to kind of try and take some of these principles and show you how we applied them in salmon. And the first example is in Chinook salmon from Western Alaska, which actually turn out to have genetic differentiation on the level of what you'd expect in a marine species. And so Chinook salmon are a keystone species in the kind of AYK region. They're a vital subsistence resource. Large declines in 3 or what we call 4 major drainages. And the lack of population structure inhibits genetic-based management. So our goal here was to try and use genomics to improve resolution of population structure and facilitate mixed stock analysis. And anyone that knows about salmon genetics will understand, but I can explain it here, is that mixed stock analysis is kind of one of the vital tools in salmon genetics. And what that is we go out here to the Eastern Bering Sea, we take a sample of salmon, and those are a mixed stock. Those could be from really anywhere from Oregon all the way through Russia. And we use kind of statistical genetic tools and genetic baselines composed of information from each different spawning stock, and then we can assign those unknown fish back to their stock of origin. And it turns out that in the Eastern Bering Sea, especially in winter, a large portion of the fish caught in this area are from Western Alaska or AYK stocks. So that's the blue here. But the thing is, we can't differentiate these different stocks into multiple pieces. And so it's kind of 4 major drainages in these areas, the Nushagak in Bristol Bay, Kuskokwim, Yukon, and then up in Norton Sound. And if we take a look at the genetic tree here, just based on 43 SNPs, which is the genetic baseline that was used before, we can see that the branches are very, very short. And so this is, I forget if this is maybe Alaska Peninsula or something that I forget, but this is kind of what we're more used to in salmon, with these long branches and hierarchical structure among drainages. But in Western Alaska, they're just extremely, extremely short branches, and these drainages are very difficult to tell apart. Hopefully I have, so we need genomics and adaptive loci. So why is this? Well, if anyone who's flown over Western Alaska before, it's probably going to make quite a bit of sense. It's a really flat tundra, boggy environment, and this has facilitated large kind of river capture events in the past. So this is the Auke Delta, and the Yukon and Kuskokwim come within I don't know what it is, 20, 30 kilometers of each other, I think, and the only thing that separates them is kind of this like boggy tundra environment. So what's happened is, because these rivers have changed paths so many times in the past, let's say 10,000 years, there's been a lot of opportunities for gene flow among these populations, and you actually get levels of genetic structure on the order of what you'd see in a marine fish. So yeah, levels of gene flow similar to marine systems. Okay, so we wanted to try and crack this nut, and we use what's called restriction site associated DNA sequencing. I forget if I have the slide on it. And so we sequenced about 50 individuals from representative populations in Western Alaska, and then one up in the upper Yukon is kind of an outgroup there. And before I get into this, I just want to talk about kind of what a genetic marker actually is, or the genetic markers we use. The genetic markers we use are called SNPs, which is a single nucleotide polymorphism. It's a single base pair change in the DNA sequence. And this is what allows us to kind of understand population differentiation and diversity based on the different alleles or different bases at these different SNPs. So when I say SNP throughout the rest of the talk, that's what it is. And rad sequencing is a way to kind of subset the genome down into a manageable number of pieces that we can sequence. So we use a restriction enzyme to fragment the genome at specific places that match a certain DNA sequence. And then we sequence 100 base pairs, or now it's 150, but back in the day it was 100, around each one of these restriction sites. And then we assemble these into what's called stacks of similar sequences, and then call SNP phenotypes using the sequence count. Okay, so this is just a look at the genetic differentiation of all the different SNPs that we identified in this region. And what you can see is that most of the SNPs are showing extremely low genetic differentiation. This is FST, so this is ranges from 0 to 1, and values near 0 have pretty low differentiation, and values anything above 0.1 or 0.2 is really pretty high. And so you can see we found a lot of SNPs that show very low nuclear structure, but we also found these really high FST SNPs that are probably putatively adaptive. And so as far as the power of genomics loci, when you combine, or of genomics and adaptive loci, when you combine all these 10,000 SNPs together and you look at population structure in the AYK, you can actually see that these things are starting to pull apart, whereas with the 43 SNPs, you really don't see any structure at all. So these are two PCAs. This one's each dot is an individual, the different colors of different populations. And with the 43 SNPs, it's essentially a shotgun blast, and there's really no kind of detectable or usable genetic differentiation among these three populations. But as you start to add more loci with the rad sequencing, you can actually see that sort of three different clusters start to pull apart, and you can actually start to get these populations to differentiate. And so the next step here was, we saw that we could differentiate these things with 10,000 loci. We wanted to develop a panel of high resolution, using just the high resolution SNP, so that we could screen that on thousands of individuals. And so we use what now is an antiquated chemistry called fluidigm. But what you do now is you develop what's called a GT-seq panel, and you'd pick the top 500 or so of these SNPs, and you develop PCR primers for them, and then you'd conduct amplicon sequencing on a MiSeq or something like that, and then you could you can genotype a lot of individuals really affordably at these high resolution SNPs. So again, Western Alaska was previously one reporting group for mixed stock analysis. That means that we could for all these different populations, we couldn't differentiate any of them when we caught salmon that we didn't know their origin, and so they all had to be assigned back to this Western Alaska reporting group. But with these rad-derived SNPs, 3 reporting groups are now feasible, so we can actually differentiate Norton Sound, the Lower Yukon, and then Kuskokwim and Bristol Bay still make one reporting unit because they're just really tough to differentiate. And these new SNPs are actually being integrated into management by ADF&G and our lab. So there's a lot of different, research cruises and bycatch samples in the Eastern Bering Sea, and we're integrating these SNPs into that management protocol. So just to zoom out, we identified, top high-resolution FSP SNPs that are putatively adaptive, and we leveraged those SNPs to differentiate populations that we couldn't tell apart before, and we're now kind of using that to inform management and of these AYK Chinook salmon populations, which are, a really important topic right now. All right, so that sort of wraps up the Chinook salmon portion here, and I'm doing kind of good. And now we're going to transition into Sockeye salmon, and so the second half of my PhD, and then continuing, on to present, is trying to understand ecotypic diversity in Sockeye salmon across Alaska. But before we get into that, I want to talk a little bit about how we actually identify these adaptive loci and adaptive regions. So what I showed you before, as far as that plot with all the SNPs kind of going from low to high FSP, is pretty much how we identified adaptive loci before we had genomic resources. So in other words, we genotyped 10,000 SNPs. We'd look to see which ones had high FSP, and we'd use these different programs to kind of draw an artificial line of significance. Anything above that is putatively under divergent selection, meaning putatively adaptive. So in other words, if something's diverging and it's more differentiated than we'd expect under neutral processes, we'd expect it to be under divergent selection, and we call it sort of a putative adaptive locus. The problem here is we don't actually know where these SNPs are in the genome. So like these 2 SNPs, for example, could be right next to each other in the genome, or they could be on completely different chromosomes. And so we're kind of lacking an understanding of kind of the genomic architecture of adaptation. And so what we're trying to move towards now is, instead of just looking at these anonymous adaptive SNPs, we're trying to place them on genomic resources like linkage maps or sequence genomes, so we can try to understand exactly where the adaptive loci are found, and try and actually sort of hone in on their function. And so this is another example from COD. This is that linkage group 1 high FSP area that I showed kind of in another plot in another way. But now, based on this, we can actually start to understand the mechanism. So in COD, for example, we know that a lot of these SNPs align to chromosome 1, and there's been a lot of studies following that up that show that these SNPs are found on a large inversion, which is kind of a large piece of the gene of a chromosome that's flipped in some individuals so that they can't recombine. And essentially what it does is it protects all the genes in that region from recombination, and allows them to kind of evolve together and without being kind of broken up by recombination. So we're moving from this kind of rudimentary understanding of, okay, yeah, there's some loci that show the high FSP, and they're probably adaptive to, they're found here in the genome. They're caused by this, genetic architecture, and these genes are found in this region. And taking this another step further, the ultimate goal here is to try and understand specific genes involved in adaptive divergence. And this is a really nice example. This is a paper that just came out in Science on GREB1L, which is a gene, the GREB1L region, which is a gene involved in run timing in Chinook salmon and steelhead. And you can see, so here's where the region of high association is. It's on chromosome 28 here. And you can actually see, so here's where the genes are, and here's where the region of high association is. And you can see that that region's kind of towards the tail end of GREB1L and kind of in the intergenic region there between these 2 genes. So this is kind of our ultimate goal here, is try and find these genes that are involved in adaptive divergence. Okay, so can we find these genes or genomic islands of kind of conserved architecture in Sockeye salmon ecotypes? All right, so a little bit of background on the Sockeye here. Most of my work was done in Bristol Bay, which is in the Bering Sea there, in the Eastern Bering Sea. And Sockeye returned to 9 major rivers in this region. And each one of these rivers has many different populations, which a lot of them are also genetically differentiated. And so the important piece here for kind of our work is that Sockeye salmon have different biological features depending on the habitats they occupy, leading to the formation of distinct ecotypes. And the most kind of extreme and conspicuous example is Sockeye that spawn on these lake beaches versus ones that spawn on these small streams. So lake beaches are pretty deep. You're talking, 1 to 3 meters maybe. Each one of these different circles is a nest. So a different pair of Sockeye use those to spawn. And in a lake environment, sexual selection favors large, deep-bodied males. Whereas if we move to the streams here, so there's these really small streams with deltas that can be only a few inches deep, and then streams themselves that are maybe only 6 inches to a foot deep. And these streams have very high bear predation. And the larger you are, the more likely you're going to get eaten by a bear. So there's extremely strong selection on these much smaller, more fusiform phenotypes in the streams. And I think, let's see, I should have a picture of some of those ecotypes after this slide. So the study system here is the Wood River. It's a series of 5 kind of lakes stacked on top of each other in northwest Bristol Bay. About 3 million sockeye return to the system annually. And the really important component of this system for our research is there's 3 spawning ecotypes: beaches, rivers, and streams. And so again, just to kind of circle back on what I was talking about a couple of slides ago, this is a representative male from each ecotype, and then the environments that they spawn in. And so again, the most conspicuous phenotypic difference is these beaches, which are these these big, deep-bodied males, versus streams, which is kind of a much smaller fusiform body type. And the rivers look more like the beaches. There's not really that many depth constrictions there, but we know that there's a lot of other different kind of adaptive differences between river populations and the other populations. Okay, so as far as the study design for this first part here, we looked at 6 populations, 2 from each ecotype. The first and second lake of the system make kind of a nice replicated stack design here. And so we were able to get a river, a stream, and a beach from each of these first 2 lakes, about 50 individuals per population. And then we did rad sequencing, and we aligned about 3,500 SNPs to a linkage map. And again, a linkage map is just a way that we can visualize genetic markers across the genome. And so here we are getting into the results. And what we see is that, like the other species like cod and things like that we've been looking at before here, most of the loci show very low differentiation. So again, each different dot here is a different SNP, and the black SNPs are what we classified as neutral, and they show very low differentiation. For geneticists in the audience, the overall FST in the system is about 0.01. So on the order of a marine system, maybe a little bit higher, but not too much. But the really interesting thing here is that there's a couple areas of the genome that show extremely high differentiation. We call these genomic islands of divergence. And the largest ones, are the most conspicuous anyway, are on chromosome 12 here and chromosome 13 here. So kind of same thing we've been seeing in cod, where there's very low neutral structure across most of the genome, but a couple areas that are likely involved in adaptive divergence as these ecotypes differentiate. Okay, so just kind of some take-homes from this Wood River rad sequencing. Low neutral structure across most of the genome, and the structure is largely driven by genetic drift. But there's these large islands of genetic divergence that show high levels of differentiation among ecotypes. And so I thought this was pretty neat, and we were excited to find this in one system. But what we really wanted to kind of get into next is try and understand if these islands show similar patterns in other systems. In other words, are similar genetic mechanisms responsible for phenotypic differentiation across multiple colonization events? And so what we did is we looked at these islands, and we developed 7 high throughput assays for SNPs in these islands. They were TaqMan assays. We wouldn't do it that way now, but we did it that way then. And then we genotyped these TaqMan assays on 32 different populations, which encompassed a large suite of ecotypic diversity. And so the Wood River's here, Iliamna, which is a couple drainages over in Bristol Bay here, Chinook, which actually flows down south into the North Pacific, not into the Bering Sea, is over here. And then the Copper River, which is pretty genetically different, flows into Prince Sound, is over here. And so I will say we used neutral SNPs genotyped by the Alaska Department of Fish and Game and all these populations to kind of give us a baseline for what looks like neutral SNPs and then what our island SNPs would look like. And so this is just another one of these plots that I've been showing quite a bit with genetic differentiation across the genome. Our neutral SNPs down here, and our outlier SNPs genotype with TACMAN here. And this is from the Wood River, so there's no surprise that when we re-genotype these fish, we saw the same pattern. But where it starts to get really interesting is we saw that in Iliamna, which is a totally different system, we see the same exact pattern with the same islands of divergence kind of showing up. Same thing in Chinook when we look in another system, and then the same thing in the Copper River, although it's a little bit more confusing here because neutral structure is higher, but you can still see that chromosome 12 and chromosome 13 still have these high differentiation islands. Okay, so just kind of zooming out and looking at the genetic structure of these loci. So structure of the neutral SNPs is driven by drainage, and this is exactly what you'd expect in salmon. Salmon shows some of the strongest, hierarchical structure of any fish. Each different color here is a different drainage, and so at neutral SNPs, things are grouping first by drainage, and there's not really much genetic differentiation by ecotype that's evident from this tree. But if we look over here to the island SNPs, things absolutely aren't grouping by drainage. There's sort of there's populations from most drainages that are showing up in the different nodes. So if you think about up here, so this is Chinook, this is Quijack Iliamna, and this is Copper, this is Wood. And for this area of the tree, that all the drainages are represented. So in other words, there's no real rhyme or reason to the structure at these island SNPs. They actually don't really differentiate by ecotypes, with the exception of up here. And they don't really differentiate by drainage. So, what's going on here? Why are these SNPs, not showing patterns that are associated with ecotypes or by drainages? So, where did we go after that? So we genotyped these 30 populations with, a small subset of these SNPs we discovered with rad sequencing. And we're starting to see some interesting things where these SNPs look really adaptive across a lot of different systems, but we don't really know exactly what's going on, much more than that. So what do we know? So we know SNPs and islands of divergence show high differentiation across drainages. And what we don't know a lot. We don't know the full landscape of adaptive divergence. We don't know the genetic mechanisms causing this differentiation, and we don't know the genes involved in the differentiation. And so to try and kind of get a better idea of these, we started to use genome resequencing, which means that we moved from about 3,500 SNPs to over 2 million. And I will say, so this has kind of been in the past 4 years when it went to, sort of mostly me leading this charge to mostly other people leading it, which has been really cool to watch. So first off, I want to thank Kristen, who is a total whiz in the lab, who got this whole lab side of things figured out, and then 2 postdocs, UA and Peter, who have passed me analytically and then some by lot. So it's really great to have this team. And so we looked at 7 different populations with this genome resequencing in 3 different drainages. Back to the Wood River system here, and then over to Iliamna, and then we genotyped a Sea River population, which is kind of an outgroup. And if we just look at a PCA of all the SNPs, no real surprises here. Everything groups very hierarchically. So Iliamna, all the Iliamna fish go in here, all the Wood River fish go in here, and then the Sea River one is kind of an intermediate, just as we expect. And again, hierarchical structure, neutral FST within drainages is about 0.01. We knew this with rad sequencing, we knew this with neutral SNPs. There's no surprises here. But where it gets really surprising is when we actually start to look at the heterogeneity of differentiation across the genome. And so these are Manhattan plots with all 2 million SNPs for the Iliamna drainage and the Wood River drainage. And we can see again that there's most of the genome is showing relatively low FST, and then there's these kind of blue and red spots that are extremely highly differentiated. And it's interesting, we can look and we can see, some of them are common among the drainages and some of them aren't. So a good example there is like chromosome 22 actually is really, the differentiation is only really high in the wood, not really in ileomna. But there's 4 regions that are highly conserved across both drainages. That's chromosome 3, chromosome 5, and then most notably chromosome 12 and chromosome 13, which is the ones that we also saw in the rad sequencing. Just a quick kind of zoom out to think about the differences between rad and whole genome. And this is for folks that might be trying to plan genetic studies in the future. The resolution that you get with rad is way, or maybe I'll put it this way, the resolution you get with whole genome is way higher than you get with rad. And so with rad, we started to see that there's definitely some islands on linkage group 12 and linkage group 13. We probably picked this one up on chromosome 7 here, but we really missed the one on chromosome 3, we missed the one on chromosome 5, missed the one on chromosome 22, missed the one on chromosome 15. Actually turns out that this is flipped and this island is right around the MHC, which is kind of cool. Anyway, we missed a huge amount. And so just a quick kind of like comparison of cost versus what data you actually get. So rad sequencing, you can budget it's maybe around $50 a sample. And this whole genome, we went to about 7x coverage, which is probably higher than we'd go in the future. That was pretty expensive. But if you go to more like a 2 or 3x coverage, you're probably talking about $100 a sample. So, give or take, whole genome sequencing costs about twice what rad costs, at least in kind of our calculations. And when you're looking for kind of signatures of adaptation, I think it gets you a lot more than twice the data. So, rad sequencing is a really good first pass for understanding kind of population structure, and you can get a really good look at kind of neutral structure and some peaks at adaptive loci. But whole genome has just really revolutionized the way we've been able to understand what's going on in Sockeye. And so another really good example of that is in chromosome 13, which is one of the bigger islands that we found. And so this is what we had with rad sequencing. We were psyched at this. We found 6 loci within 500 kb, which we were honestly really lucky. But with whole genome, we've got hundreds in that region. And what you can actually see is that, so notice the difference in magnitude of FST here. So the highest SNPs we had were 0.3 here, whereas here we have a 0.6. And so it actually turns out that this little peak was probably somewhere in here, and what we're actually looking at as the real peak is over here. And the real peak, I won't show all this, but the real peak is actually only about 20 or 30,000 base pairs across, whereas with the rad we thought it was 500, and we really didn't have a good idea. All that we knew is that the peak started before this locus and it ended before this locus, which is actually over a megabase. So with rad we thought it was potentially a million base pairs wide. With whole genome, we figured out that the target of selection most probably is 20,000 to 30,000 base pairs. So what we can do, we can actually look at the target of selection versus just this big region of linkage. We can actually try and understand the mechanism of inheritance. You'll see here, I'll show that in a second in the next slide. And we can actually look at the functional importance of the regions. And so one thing that's pretty neat with this whole genome data is we can start to look at the structure of each of these islands of divergence. So we can see this one's for chromosome 12. And it turns out that this looks like it's probably an inversion that's about 60,000 base pairs across. This is homozygote 1 for the inversion, 2, and then these are heterozygotes. And there's also some kind of additional variation within the inversion. And the main thing here is that beach and sea river types are all found they kind of have the A1 primary allele of this inversion, whereas tributaries and island beaches are over here. So we can actually start to see, which populations these things break apart and which SNPs differentiate them. And chromosome 13 in particular is really interesting. So For chromosome 13, we actually don't think it's an inversion because there's 3 primary alleles. So this is homozygote allele 1, allele 2, and allele 3, and then the heterozygote in between. And another really interesting piece of this is that I didn't put it anywhere, but there's an estrogen receptor gene that's kind of right smack in the middle of the kind of super conserved region on chromosome 13. So we think that the actual target of selection is probably that gene. Another interesting thing here is that there's really no rhyme or reason to which populations are being grouped here. So again, the mainland beaches, are kind of grouped over here, kind of like chromosome 12. But the Agulawak River is extremely unique, kind of with its own allele over here. And then in A3 is the island beaches, and then some of the streams. So I only showed 2 of these islands, but we've looked at a dozen plus, and what you see is there's actually no consistent signal among islands. So every island kind of does a different thing. In other words, like the chromosome 12 island seems like it differentiates beaches from the rest of the population. The chromosome 13 island, sort of beaches and maybe large rivers. And then if you start looking at more, you see that there's just different sets of populations are differentiated by different islands. And so there's kind of this whole mosaic of adaptive differentiation. It's not really like all the islands are doing the same thing. Okay, so what are some conclusions here? So adaptive divergence in Sockeye appears to be largely controlled by a small number of genomic regions, which I will say, which are also within themselves small. So 60,000 bases wide maximum, which is really small for this type of thing. We think these genomic regions represent kind of evolutionary shortcuts that are used repeatedly to facilitate adaptive divergence with gene flow. So as these Sockeye salmon colonize new systems, we think that these regions are kind of selected on, and different combinations of different alleles of these regions lead to the kind of a large amount of the phenotypic differentiation that we see within these Sockeye. The other thing that's really interesting is that these predefined ecotypes are not always grouped together, and the diversity is kind of a gradient, and it's also really cryptic. So in other words, if you just look at 2 populations, you can't really have a good idea of what they're going to look like adaptively. So yeah, so genetics is bringing kind of another tool to the table that you wouldn't be able to understand, just with neutral structure or with kind of observation. And the mechanisms that underlie these regions likely vary and include structural variants like inversions, for example, in chromosome 12. Okay, so I'm trying to think about kind of the conservation implications of this, within and outside of Sockeye. And so in Sockeye, adaptive differentiation is not correlated to neutral differentiation, and adaptive regions represent kind of a mosaic of diversity rather than a single pattern. So managing based on neutral diversity would be kind of, or a single adaptive genomic region would be imperfect. So if you manage this on neutral diversity, you kind of lose all this kind of ecotypic diversity underlying that neutral diversity. And if you manage it on just one of these regions, you'd be ignoring, the maybe dozen or so others that are kind of showing different patterns. So, the best way would be to try and preserve the full landscape of adaptive divergence, including this cryptic diversity, to ensure that adaptability and resilience is maximized. And kind of a zoom out to other species here is that, genome resequencing will facilitate informed conservation of adaptive diversity in many species. Okay, looks like I'm doing okay for time, but I still have a few more things to talk about. So that's kind of the end of the salmon section of the talk here. I wanted to talk a little bit about what we're doing at the Alaska Fisheries Science Center as far as genetics research. So one of our big areas is going to be population genomics of managed marine species. Talking about things like pollock, Pacific cod, sablefish, rockfish, and things like that. So genomics work at ABL will focus on understanding adaptive diversity of managed species across environmental gradients. Primary species, things like I said, sablefish, peacod, pollock, etc. And we're really interested in compiling genomic data from multiple species to explain similarities, i.e. similar responses to environmental breaks, and differences that might be mediated by life history. One area kind of in particular that's really interesting is the Bering Sea versus the Gulf of Alaska, and kind of understanding how that large environmental gradient plays out adaptively in these species. And I just want to say that I think it's going to be really interesting to compare the halibut data that you guys are generating with data from some of our other fish like peacock and sablefish to try and see if we see kind of these similarities in what we can use that for. Okay, last thing here before I wrap up. I want to talk a little bit about some of the work we're doing on eDNA. And so, you know, we're spending a pretty good amount of time trying to pilot eDNA and trying to build our eDNA program at Alaska Fisheries Science Center, because we think it's going to be a really valuable tool that can kind of augment traditional survey data. And so just for folks that might not be super familiar with it, the sort of main approach here is you go out and you collect a water sample, you vacuum filter it, you preserve that filter, and then you can do kind of two main things. You can either use qPCR to detect a single species or a few species, which is higher resolution for that single species, but you only get data on sort of a single species. What we're more focused on here is this technique called metabarcoding, and it detects many species using high throughput sequencing. And then primers can be targeted for different taxa, so things like fish, mammals, algae, phytoplankton. So you can really get a kind of a full community, view from this metabarcoding. And so our first eDNA study was in this lake in Michigan, and we went out and we set traditional gear, so gill nets and spike nets, and then we took eDNA samples right before we set the nets. And what we found is that with the traditional gear, we got 12 species total, but with the eDNA, we got 40. And we were actually able, I don't know if I put in this stock down, but we were actually able to pick out a fine scale spatial heterogeneity in fish communities using eDNA that was absolutely not evident in the traditional gear. So I think it was that, like, you know, an area like this was kind of dominated by centrarchids, whereas another area was not. And we actually picked that up with the eDNA, which is pretty cool. So it looks like it can pick up these fine scale spatial differences, which is neat. And as far as what we're trying to start here at ABL, we went out this summer, fall, and we took samples from a bunch of nearshore environments at high tide and low tide, kind of out the road in Juneau. And we just got some results back a couple days ago. We took a quick peek, and the good thing about it is it looks like it's kind of, you know, showing the results we expect. We haven't done anything quantitative yet, but you know, we found things like salmon, kelp greenling, herring, of course, starry flounder and sculpin. If you didn't find these two, you know, something would be wrong. So yeah, it's looking good so far. Another thing that we're really excited about is we're working with some folks at Oregon State to help pilot an eDNA autosampler, which is essentially kind of a series of bags that can preserve eDNA samples. And this autosampler was designed for kind of stream side use. So you'd like put it next to a stream, you'd put a pipe in the stream, and then it would take 24 samples. But we're piloting it for use in the marine environment, kind of on a pier this spring. And the ultimate goal is to try and develop a version that would be submergible underneath a buoy or, you know, a shallow bottom that could take, you know, 24 more eDNA samples, and we could come back and, you know, get those a couple weeks later. And, you know, obviously with eDNA, like any traditional sampling, you've got to physically be there to take the samples right now. But an autosampler like this would really allow us to cover a huge amount of territory and get a much better look at kind of temporal resolution. A lot of our primary assessed species have nearshore juvenile life history phases, and you know, we don't know a lot about that. So using kind of a network of these autosamplers to try and understand, you know, presence, absence, distribution, phenology of these things would be really useful. So that's kind of a long-term goal here. So future eDNA applications, monitoring nearshore communities and tracking managed species, investigating invertebrate and fish communities in untolerable habitats, you know, piloting use on fish species used for stock assessments. And one thing that I'm really excited about is understanding community composition and migration in a changing Arctic. And, you know, Alaska is kind of just one big logistics problem. And so, you know, being able to have a network of these eDNA autosamplers rather than having to take, you know, a big boat out and set really big nets and things like that, I think it would allow us to cover a huge amount more ground and get a lot of, you know, really useful data. And yeah, we're actually working with IPHC on a pilot project looking at eDNA collected from tanks up at APU. And so we're gonna see how that goes, and we're excited about that too. All right, well, that about wraps it up. Just like anything, you know, a lot of funding and collaborators that helped with all of this research. And with that, I will take some questions. Thanks so much for listening, guys.
Thanks a lot, Wes. That was really interesting, and you covered a lot of interesting ground. I would like to remind attendees that they can put in their questions on the question tab. So please go ahead, and then Dana will read out those questions. So Dana, are there any questions so far?
There aren't right now. We can give people another moment. Or if they had questions after the talk, I'm sure they could email Dr. Larson, but there are no questions right now.
Okay, well, maybe I'll break the ice and I'll shoot a couple of questions. One was related to what you were talking about, the islands of divergence, and you made a brief mention on the importance of inversions. So one question is, do inversions always result in islands of divergence? Is that one of the mechanisms for the presence of differentiation in genomes?
So inversions can absolutely be neutral, and we found one in a species in Wisconsin. So I guess was your question more like, can inversions be neutral, or like do inversions always result in differentiation essentially? Yeah. So no, I think that they're pretty common actually. You know, it's easy to see the inversions that result in differentiation, but it's harder to detect the ones that don't. But you know, if you see this big conspicuous FST peak, yeah, it's really easy to identify them.
Yeah, because I thought you mentioned that in chromosome 12 there was an inversion there. Is that correct?
Yeah. So we think so. Actually, totally confirming that something's an inversion is not super easy without long read sequencing. But we did do some analysis that makes it look like it is. So chromosome 13 or chromosome 12 has kind of two, discrete alleles, and it looks like a really strong linkage break where it's sort of like it's associated and then it's not. It's like you can't see my hands, but it looks like a kind of a rectangle. Chromosome 13, we don't think it's an inversion because it has 3 alleles, and the linkage around it is really messy. So you can see sort of like different linkage patterns and things like that. So, we're not 100% sure. We think that probably chromosome 3 and chromosome 12 are inversions, and chromosome 5 and 13 may not be. But we might not be fully sure until we do some long read sequencing.
Okay. Dana, are there any questions?
Yep. We have one question from Ian Stewart. He says, well, there's a comment and a question. He says, thanks for a great talk. The Sockeye and those systems were just as cool when I worked on them 20 years ago, but the genetic tools were pretty primitive. In highly mixed marine systems lacking physical population structure, such as drainages or habitats, which criteria might be helpful to identify the resolution that is appropriate to define important adaptive units?
That's a good question. So that's great you worked up there. That's awesome. I mean, so Sockeye in these small systems, it almost is a little bit marine-like, because there's so many opportunities for gene flow, and I think that's why why there's so much interesting genetic architecture. I guess in marine systems, I would really default to whole genome sequencing if possible in the future. Yeah, unless you really want to look at a lot of populations. And I guess I'm not sure if the question is the tool or like the cutoff for saying whether or not it's important. I don't know if you can talk or type in.
Let me see if we can unmute Ian.
Okay. Like essentially whether it's like you're curious about sort of the decision tree of tools or the decision tree of like, you know, once you find some stuff, what you might want to do management-wise.
Ian, you should be able to talk now. I'm not sure why it's not working.
No worries, but
he said the cutoff particularly.
Okay. Yeah, you know, that's a really tough question. We're really at the infancy of integrating these adaptive loci into management. And so, you know, the cop out, but I think honest answer is the rules are really still being written. You know, Sockeye in Bristol Bay, you know, they're kind of managed the way that they're going to be managed. They're managed on pretty, you know, on a resolution by drainage. And it's really not feasible to do it any other way. But for something like, let's say, halibut, you know, the cutoff for designing these management units, you know, could be different. You could change the management units feasibly. You know, I think if you found a, you know, a really different adaptive signature in the Gulf of Alaska versus the Bering Sea, you know, there might be a reason to kind of reevaluate that. Although I think that the genetic structure already shows differences between those two areas. The total cop-out answer is, show me what you got and I'll show you what I think. I'll tell you what I think. Because I guess it's a moot point talking about it if there's no no big adaptive differences. But if there are, thinking about how you might change management would be pretty important. Cop out, sorry.
Ian says thanks. And we have one more comment, not a question, but Dr. Tim Lohr says eDNA, an eDNA auto sampler, that would be cool as all get out. So.
Oh, I should say that it's also really cheap. That's the whole like point of it. Those things are usually, you know, hundreds of thousands of dollars. And this one, you know, the goal is to have it be around $5,000. So an affordable, attainable eDNA auto sampler is what I should say.
Wes, could you clarify what that auto sampler does? Does it actually filter the water samples, and then provides, you know, filters with DNA attached to it for a total of 24 samples?
Yeah, that's exactly right. So it filters and preserves the samples, and then you just go back and kind of take out the cartridges. Yeah.
Yeah, that's really cool.
Yeah, we're super excited about that. Can't wait to. They're building our first two right now, and we should get our hands on them, you know, hopefully in January or early February, and get them deployed on a dock out here next spring.
And the 24 sample capability, that's just to be able to sample over time?
Yeah, so there's a kind of a programmable interface that you could sample, like, you know, once every hour, once every day, once every week, something like that.
All right.
There's a question about PTA.
Dana, do we have any other questions?
Yeah, there's one about the PTA here.
Yeah, I'll read it aloud. It's from Claude Dykstra. For this type of PCA analysis, can you highlight different genomic structure if you bin your animals by another feature, such as longer fish or faster growers, as opposed to their geographic basin?
Yeah, it's a good question. It's actually tough. So all these different attributes co-vary, which makes it really tough. So, but like, okay, for example, if you did that on chromosome 12 right here, so this cloud is the fat fish, basically, and this cloud is not all but largely. So the yellow population is like the really skinny type, and then the the pink and the blue is like the really fat type. So for this one in particular, it would look like those two are differentiated. This one would be a bit different. So this one, these are the fat type with some skinny types in here. This is the fat river type, which is different than the fat beach type. And then this is the stream, which is the skinny ones, but then there's also island beach, which is sort of fat. So it gets really complicated. Yeah, the short answer is that phenotype and ecotype covary to the point where we've gone down this like genome-wide association with different morphological features route before, and we've just sort of like spun ourselves in this circle of like, well, all these phenotypic characteristics are associated with ecotype to the point where we should just make the ecotype kind of a categorical variable. Yeah, so again, sorry, Claude, cop out answer. But I will say that, like, geographic, basin is not what explains this, which is, I think, probably super clear. So the pink and the blue, those are from different basins. The yellow and the orange, those are from different basins. So whereas there's hierarchical structure by basin with the neutral SNP, the SNPs here, it's nothing by basin. And actually, so we've looked as far south as like the Fraser and the Columbia with some other data, and even at that level, the basin doesn't explain this. They pop into these different allelic categories.
Okay, that's it for questions that came in online. Oh wait, I think one just came in. One moment. Okay, this is from Nicole Fredrickson. She says, you mentioned that adaptive differentiation will support the conservation of many species. I'm particularly interested in Pacific herring. I work as a biologist for First Nations in BC, and they have been very interested in genetic differentiation and stock dynamics of herring in the Salish Sea. Their traditional knowledge indicates historically there were many different stocklets in the Salish Sea, but most of the modern genetic work to date has indicated that there is no genetic differentiation of the stock. So it is managed as one aggregate stock. Are you aware of any work currently underway for Pacific herring? Do you think this work could be applied to Pacific herring?
Oh, what a great question. You just teed me up right there. That's fantastic. Okay, so hopefully you know Eleni Petro and Lawrence Hauser at UW. If you don't, my email is here. My email is right here. And oh no, sorry. Yeah, if you don't know them, shoot me an email and I'll connect you guys. So Eleni just finished her PhD on this topic, and you're 100% right. I should use this example in talks. So neutral structure with microsatellites showed very little differentiation of herring, but as Eleni started to look with RAD, and I think they're trying to go to whole genome, she found that there's high genetic structure associated with temporal isolation. So herring might occupy the same environment or the same habitats, but they're temporally segregated, and there's actually some inversions that seem to be highly differentiated across those temporal groups. As far as kind of micro scale stock structure within those groups, I think there could be some, but no one's done the whole genome yet. We're working on a bit of herring up here. Hopefully, we're trying to get some stuff off the ground, but that wouldn't be BC. But you should definitely get in contact with Eleni and Lawrence and kind of zooming out. I think herring is a great example of where this work could be really, really useful. So thanks for asking that question. I hope that helps.
Okay, that's all the questions I have for you. Thank you very much.
Yeah, thanks so much, you guys.
Thanks very much, Wes. And thanks all the attendees for attending, and we'll keep you posted on future seminars. So thanks very much again, Wes. That was a great seminar.
Sure, thank you. Take care, guys.
Bye-bye.